Good evening, good afternoon, and good morning. This is the 1817th day of continuous webinar conducted by International Forum for Promoting Homeopathy. Today, our guest is Dr. Suraj Singh Bhadoria, and he is an assistant professor, Department of Homeopathy Pharmacy, Paul, Institute of Homeopathy and Research, Vadodara, Gujarat, and has made so many presentations in this forum. And today, Dr. will be discussing about recent advancement in homeopathic pharmacy. So we proudly welcome you, sir. You can begin the session. And if you want, if you can share yourself, you can share. Otherwise, Dr. Roy Thomas will share the screen.
Okay, sir.
Can you share it?
Just yes, sir. I am able to share this, sir.
Okay, then you can share.
Is it visible, sir?
Yes, visible, visible.
Okay. Now this slide is visible, sir. Pharmacy.
Yes, yes, pharmacy is
Okay, sir. So basically, today my topic is homeopathic pharmacy in the present scenario. This is very important to understand the importance of pharmacy, not only in the field of homeopathy, for every medical science and the Ayurveda, the Unani, and the modern pharmacology. Because this pharmacy is not only the subject of first year BHMS, it is a huge branch, and it should be a separate branch like B Farm, M Farm, and a separate PhD course, so that we can facilitate the complete knowledge of drug along with the complete medical science. So what is pharmacy? So it is a complete knowledge of drug in sense of the identification, selection, collection, preservation, analyzing, preparation, standardization, drug proving, and drug dispensing. So it is the complete knowledge of drug. Is it visible to all, sir? Is it visible to all, sir?
Yes, everything is fine. You can,
Okay.
screen is
Okay.
visible.
Okay, sir. So basically, in identification, we are going to identifying the alkaloids. We are going to identifying the markers in our drug substances, because ultimately identification plays a major role. Whenever we talk about the holistic approach of drug or any raw material or herbs, so the active components and the metabolites plays a major role, because they are going to show their efficacy and the therapeutic value. So the identification part plays a major role. Whereas in the selection criteria, we are using certain parts from the plants, from the animals, from the nosode, where we prepare the drug and standardize it. So the selection part again plays a major role. Collection and analyzing, these are again the main pillar of this pharmacy and the drug substances, where we collect the part and we synthesize it, we analyze their compound, we analyze and segregate their primary metabolite and the secondary metabolite, and we distinguish it on the basis of qualitative and the quantitative analysis. So the analysis part plays a major role. The preservation, here we preserve the alkaloidal property. Preparation, we have old method and the new method for the preparation of drug substances. In new method, we have the maceration and the percolation. So this again plays a major role in the preparation part. Standardization, again, it is a very important aspect for the drug quality control. Standardization means the quality assessment of any raw material, any medicinal product, or the remedy. So standardization again plays a major role. Drug proving, drug dispensing, again these are the pillar of pharmacy. So here, what are the recent advancement? Now the recent advancement is, when we talk about general aspect of pharmacy, so when there is a production, preparation, dispensing, reviewing, monitoring of medication, so it always resemble with the safety and efficacy and the use of that particular medicine in a right propagation with the quantitative and the qualitative analysis with respect to the health science and the pharmaceutical and pharmaceutical branch and the natural sciences. So the advancement plays a major role in the production unit, in the formulation of various drug substances and the external applications, the dispensing of medicines, and reviewing the medication part of the material. So this is very important for the growth and recent advancement in the field of pharmacy. When we talk about the professional practice, so when it is clinical ground, so the efficacy of drug material again plays a major role. So we have two types of proving, one is preclinical and another one is the clinical proving. So in the preclinical proving we are going for the animal study, in vitro study, cell line study, whereas in the clinical study we have a patient orientation program. So this plays a major role for the advancement in the field of pharmacy. And again, when we talk about the certain settings of the pharmacy practices, so it divides and categorizes into the community or the institutional pharmacy. The institutional pharmacy, it always deals with the setup of pharmacy in certain institution, the university and the colleges which are affiliated for the BHMS and the pharmacy courses. So these are the institutional pharmacy. So where is advancement? Suppose if we have a university where we run BHMS and the pharmacy courses, allied sciences. So when we are using certain external application, when we establish a unit for the production of certain external application like a face cream, hair oil, shampoo, dye, so this plays a advancement in the field of pharmacy practices because these are the application part. Whereas we when we talk about the community pharmacy, so it always deals with the bridging and the interlinking of the pharmacist. So when there is a whole interlink and the association of pharmacists, so definitely there is an exchange program of the knowledge and the skills. So this is a very important part for the general aspect of pharmacy. And again, it is important for the patient care in the community of institutional pharmacies, which considered as a clinical pharmacy. Now, identification. As we know, we identify the various drug material from plants, vegetables, animals, nosodes, sarcodes, synthetic, imponderabilia. So in the identification, when we talk about hybrid plants, so in terms of hybridization, when we have a collection of drug material from the hybrid plant, so it is one of the recent advancement because again it is not written in any textbook of Mandal and DD Banerjee. So where is the advancement? Suppose if we talk about the hybrid variety, when it mixes with the A compound with the B compound, when it forms AB, so this is a hybridization. So when we have the collection of drug material from such species of plant, the hybrid plant, so it plays a major role in the cross pollination and the various varieties of plant and the mixture of plant and giving a certain different varieties. So this is the identification part which plays a major role in the field of advancement in the pharmacy. Now, the selection of drug material. We have different parameters for the drug substances like genetic diversity, genomic mapping, gene pyramiding, hybrid purity, parent line identification, and DNA fingerprinting. So these all are the main pillars of molecular markers, because ultimately in our homeopathy we are always talk about the holistic approach of the drug collection, identification, selection, standardization, but we have no any peculiarity and the specificity in terms of selection, in terms of identifying drug substance, in terms of standardization, in terms of molecular marker. So when we have this type of practices in the homeopathic pharmacy, because it is very common in the modern pharmacology, but when we talk about homeopathic pharmacy, so when these things and when these parameters are commonly used and practices in our homeopathic pharmacy, so definitely we will make our pharmacy as a specific and a very precise and a very scientific. Suppose if I give you one example like Thuja occidentalis mother tincture. So we use Thuja occidentalis as a whole. It is having both primary metabolite and the secondary metabolite. The active principle of Thuja occidentalis is Thujon. That is having two main categories, that is alpha Thujon and the beta Thujon. So these are the main active principle which shown their therapeutic value in the clinical or any living entity. So here we are going to isolate first Thujon from the Thuja occidentalis. Afterwards, we standardize it in compared to the Thuja occidentalis. So this is the comparative analysis. So this is just like a molecular mapping in the source, molecular clubbing, the synergistic effect. Synergistic means when A combined with B and when it forms C. So this C, the property of C is different than A and B individual. So this is the synergism. Otherwise, when it form AB, so it is a mixture, because the AB, the property, the physical and the chemical property of AB resemble with the physical and chemical property of A and B individually. So here we are not going to talk about the mixture. We are always talk about the synergistic effect of that particular molecular sciences in the various drug substances. And when we have the molecular dimensions, so definitely it is very important for the various aspect for the uh, force and the action of that particular molecule. So this is the recent advancement in the selection of drug substances. Again, it is not written in any textbook except the modern pharmacology. So that subject and that parameter should be included in our homeopathic pharmacy. Then only we can say homeopathy is now evidence based medicine. So this is very very important aspect for the homeopathic pharmacy and pharmacy in general also. Now the next is collection part. Now again it is a very important aspect. Previous aura, when we talk about the Ayurvedic system, the herbal sciences, the naturopathy. So generally they are going to collect the drug material by the prayer. So whenever they are collecting any substances from the plants and the vegetables, so first they go for the prayer, and after prayer they collect the source and the that particular part from the plant and vegetables. So what is the role of this particular prayer? It is ultimately enhancing the dynamic power of that particular source and the metabolite, the component, the active principle, the secondary metabolite, the primary metabolite. So there is an enhancement in the dynamic power of the drug substance. So again, nowadays it is not commonly seen. It is very rare because we are adopting the Western area, the modern pharmacology, the modern sciences, but we escape certain, what we can say, the classical practices which were used in our ancient time. So that should be followed and that should be carried out to maintain the dynamic influence in our drug substance. So again, this should be included in our homeopathic pharmacy also and pharmacopoeia also, so that we can have a huge number of variation in the collection of drug substances and their dynamization. When we talk about preservation, so there should be preservation of the molecular dimension. We always talk about that our drug substance should be away from the strong smelling bottle, perfumes, sunlight, damp, and some other exposure, chemical exposure. But beside of this, we should focus on the molecular dimension. We should focus on the physical and the chemical property. We should focus on the metabolite. We should focus on the therapeutic value of that particular drug substance. So this is our major aspect of the preservation of any drug material, any raw material, any herbal product, any substances. So again, it is very very important part, and it plays a very important in field of pharmacy, homeopathic pharmacy, general pharmacy, or allied sciences. When we preserve these metabolites, so we preserve their active principle, we preserve their therapeutic value. So when there is a preservation of therapeutic value, so ultimately the efficacy will be more. When there is no proper preservation of metabolites, the active principle, so they were destroyed. So there is no proper facilitation of their therapeutic value in terms of force in acceleration. And when there is no therapeutic value, so ultimately it lost the efficacy. So this plays a major role in terms of preservation. Now this is again important for us. In our homeopathic pharmacy we have only three scale. decimal scale, centesimal scale, and the 50 millisimal scale. Now, what we can do, we have to first make a different drug vehicle ratio from 1 raised to 1 to 1 raised to 10, as so on, like baseline sample, standard sample, trial sample, and the error samples. So it gives you the generalized idea and the conclusive result for explaining the various scales and the formulary of drug and medicine, because ultimately we followed only decimal, centesimal, and 50 millisimal scale. What about other drug vehicle ratio? Because recently when we enter into the in vitro study, so we found that certain drug substances shown their efficacy more in 1 raised to 1. There are some drug substances which shown their efficacy in 1 raised to 6. There are some drug substances which shown their efficacy in 1 raised to 9. So there is no homogeneity, there is no what we can say, the similarity in the drug vehicle proportion and their efficacy, because family is different, sources are different, kingdoms are different, species are different. So ultimately we should also focus on the different drug vehicle ratio. It should not always centesimal, decimal, and the 50 millisimal. So we should focus on that. Now, how we will prepare this? Now, the baseline sample is, suppose we started with 1 is to 1. So it is 1 is to 1, 1 is to 2, 1 is to 3, 4, 5, 6, 7, 8, 9, 10. So these are our baseline samples through which we started drug proving trial. We started to determine the efficacy of medicine in respect to drug and vehicle proportion. So this is a baseline sample because it is our base through which we started trialing. Next is standard sample. The standard sample are those sample which shown more efficacy. Suppose out of this drug vehicle proportion, the 1 is to 3, 1 is to 4, 1 is to 6, and the 1 is to 9. It shown more efficacy and the very good result in vitro study, in the cell line study, and in the animal model. So this is our standard sample. Now they are going to be tried. So this is our standard one. So standard means what? When they quantify and qualify in terms of standardization. Now, afterward, when we talk about trial samples, now this trial sample should be entered into the clinical variety. The previous one, the standard samples, it applies on preclinical only. The cell line model, in vitro study, and in vivo study. Whereas this trial sample, it should be applicable for the clinical variety also, other than preclinical. So same, you can see the drug vehicle ratio is 1 is to 3, 1 is to 4, 1 is to 6, and the 1 is to 9. So these are the trial samples which are suitable for that particular orientation of clinical drug proving or the homeopathic pathogenetic trial. So these trials, these samples quantify, qualify in terms of standardization, in terms of in vitro cell line study, in terms of animal study, and afterward it includes into the clinical variety. So this plays a major role. Again, it is not written in any textbook of homeopathic pharmacy, so that should be included, because ultimately when we include this part, so we can analyze more and more, we have more and more multiple option for the preparation of drug in a different ratio, in a different skills, and definitely we would conclude the generalized idea for the efficacy of drug material. Now, when we talk about potentization, again, it is not recent advancement. It is already written and it is already followed by our ancient era with the history and the development of pharmacy, homeopathic pharmacy. So, where is advancement? When we include the influence of beam, a laser beam therapy, or the synergistic inclusivity in potentization, so this will give you the general aspect and the advancement in the field of homeopathic pharmacy. Suppose if we do trituration or succussion under the mortar pestle or the electric succussion machine, potentizer machine, so there should be inclusivity of laser beam. Why? Because this laser beam, this delta H and the beam of light, it accelerates the electrons from lower shell to the higher shell and when there is a jumping of electron from lower to higher, so definitely it gain the energy. Delta is the power of delta is increases as per the Bohr's atomic theory. And when there is a increase of delta H wavelength, so there is a formation of bond in between two different or two respective atoms or two different component. And when there is a acceleration or the facilitation of electrons, so generally it gain more and more energy. And when there is a preservation of energy with the higher intensity, with the higher wavelength, so it reflects more and more efficacy in the preclinical or the clinical ground. So this is in general aspect of the potentization. So that should be also included. It is one of the experiment. You can also took this experiment in your PhD thesis, in your MD courses. Just see and try the such type of experiment with the homeopathic pharmacy. So this will give you a very new inclusivity in the potentization. Now again the space therapy. What is space therapy? Now in India we have ISRO. Now in this ISRO there are certain projects and the certain fundings given by different allied and the modern pharmacy and the applied pharmacy and the some other field of sciences, but again there is no inclusivity of homeopathy. Why not we can go for the potentization in space? So again we have a certain dimension and we have a certain conclusivity with the space therapy. So this will also give you some remarkable changes of potent the effect of potentization in space versus with the modern era. So this will be the very fruitful part for the advancement in the homeopathic pharmacy. Now the next is standardization. Now again it is very important. What is standardization? When we talk about the quality assessment of any raw material, any drug substances, any remedy, any medicine, in terms of or in respect of quantitative analysis and the qualitative analysis. So this will set to be a standardization. So standardization, so organoleptic. We have color, taste, odor, texture, fracture. So these all are the organoleptic evaluation. Whereas in the botanical ground, we have both macroscopic and the microscopic evaluation. So in the macroscopic, we have different shape, we have different arrangement of external feature, we have different marking, we have different pattern of arrangement of flowers, the arrangement of vessels on the leaflet. So these are the macroscopic, which are seen by our naked eye. And the microscopic, these are the structures and the quality assessment which are not seen by our naked eye, which are seen under the effect of microscope. That's why it is said to be a microscopic. So generally it is divided into the qualitative, the quantitative, the SEM study and the powder study. Now recently in our Parul University, we started with the SEM analysis, that is scanning electron microscope. In electron microscope, they have two main variety, the SEM and the TEM, scanning electron microscope and the transmission electron microscope. So generally this type of microscope has been used to determine the various minute and the invisible and the finest particles, just like a nanoparticles. So recently we started this SEM analysis for the nanostructure, for the nanoparticles, which is one of the recent advancement in the homeopathic pharmacy. Another one is the powder study. When we again it is recent advancement. Powder study means what? In our Parul University, we started standardization of various trituration like 1x, 2x, 3x by XRD, by TEM, by SEM. So this is what? This is the XRD defectometer technique. So in the powder study, when we talk about XRD, when we talk about SEM, when we talk about TEM, so ultimately it is used to determine the various structural pattern and the structural formulation of the given solvent system. So this is the powder studies, and it is also commonly used in the physics and the allied sciences in the field of chemistry also. But now in our Parul University, we started this type of practices with our BHMS students. So it is one of the very proudest moment for us. Being a teacher, the growth of our students plays a major role. So this is again a recent advancement. In the physical one, we have moisture content, extraction value, ash value, fluorescent analysis. These are commonly seen. These are not recent advancement, but these are very, very important for any standardization of herbal drug material, any raw material, and in any medicine. In the chemical analysis, we have HPTLC, we have GC, we have GLC, we have HPLC, we have DNA fingerprinting. So this all comes under the chemical analysis. Recently, in our PIHR, in our Parul University, in our JNHMC college, our first year student, when they prepare certain external application, so they standardize such external application with the help of HPTLC, with the help of HPLC, and with this DNA fingerprinting also. So this is one of the biggest challenge and the advancement in the field of homeopathic pharmacy, and that should be also included in each and every chapters and the syllables of our BHMS. So it will be fruitful for all. The next one is the biological evaluation. Again, in our Parul University, we started animal study, the biological model in homeopathy. So again, it is very important because people are not easily accepted this, because they thought that homeopathy drug proving it is always on the healthy provers, because we have both mentals and the physical symptoms by human beings. But ultimately, when we talk about animal study, so in animal study we determine the toxicity of that particular drug substance. So without determining the toxicity of the drug substance, we cannot be enter into the clinical ground. So this should be important part, and it is again a recent advancement in our homeopathic pharmacy, because as per the organon of medicine and the homeopathic philosophy, the animal study is not included in any section in any aphorism. So that should be included now, because if doctor animal alive, so definitely they mention about animal study, but due to some limitation we should follow that, and we should always have the theory of advancement in our organon also. So this is again important aspect. In pharmacological aspect, we have bitter, we have foaming index, swelling index, hemolytic activity. So these all are determining the pharmacological activity of the drug substance. Whereas in the microbial contamination, we have basically what we can say, aerobic and anaerobic bacilli. So again, in our Parul University, we started in vitro study. Like suppose if we prepare any sanitizer, homeopathic medicated sanitizer or homeopathic medicated oil or shampoo. So first it should be tested in the microbial assay on certain aerobic and anaerobic bacilli like Staphylococcus aureus, a gram positive, a gram negative bacilli. So these are again important for us, because ultimately through which we determine the concentration of our drug substances, for their efficacy, for their toxicity. So this is again a recent advancement in our homeopathic pharmacy. So these all are the very, very important parameters for standardizing, for quantification, for qualification, and for making our drug as a more scientific, specific, and a precise in terms of its therapeutic value. Now moving to us next slide. Now we have this drug development. Now what is drug development? Drug development, when we proceed the various composition and the molecular marker of any drug entity with the series of dilution, with the different steps and the systematic manner. So this is the drug development. So first there is a discovery, there is a development, then after discovery, after identification, after selection, after collection, we have preclinical research. In the preclinical research, we have in vitro model, we have animal model, we have microbial assay. So these all are the preclinical study, because it is before clinical. That's why it is said to be a preclinical research. Now the next one is the clinical development. The clinical development, it always define the determination of efficacy of any drug, any medicine, any raw material in the clinical setup, in the patient orientation. So this is the clinical development. So in the clinical development, we should always started with the drug proving in the multicentric, in all age group, in different places, in different geographical area, in a different sex orientation, including third genders also, not only male and female. So the proving of drug material in third gender should be also plays a major role, and it also reflects the remarkable changes in our HPT. So it should be included in our textbook also, because in our textbook we have only two gender, male and female. Where is third one? So it should be included. The regularity review, this regularity review, it gives you the holistic and the in general concept about the symptoms analysis, the signs, and the therapeutic aspect, the pathogenesis, the pathophysiology of that particular human entity with the drug proving. So this regularity review should be also included in our recent advancement. The post-marketing surveillance, it includes with the pharmacovigilance activity. When we talk about pharmacovigilance activity nowadays, we have various institutions with the different committee of pharmacovigilance. So the pharmacovigilance committee is a committee which based on the stoppage or refusing the adverse event or the side effect of any drug material after stoppaging and refusing it. So this is the pharmacovigilance. So this post marketing surveillance plays a major role in the recent advancement. So that should be also included in our drug substances, because every drug entity, every particle loses and release and lost their physical and the chemical property, their therapeutic property after certain time interval, after certain period. So that should be also resolved, that should be also surveillance and analyze in drug development. There is no stability of any product, any material, either they are in a dynamic or ultra high dilution, either they are in a crude or the raw form. So that should be also replaceable. Now this is our started, and this is our preclinical study in Parul University. We started determination of the efficacy of homeopathic medicated mustard hair oil in animal model. Now what we found, that in a particular concentration of that particular mustard oil and the mother tincture, it gives you the remarkable changes in the model of alopecia in the male rats. So this is one of the preclinical study which is conducted in our pharmacy institute, pharmacy college, Parul University, under the guidance of Dr. G.S. Chakravarti sir, who is the principal of PIPR, Parul Institute of Pharmacy and Research. Now this is the emergency handling. When we talk about recent advancement, so there should be a separate catalog of those drug material, indexing of those drug material with the respective potency, which should be used for the handling of various emergencies and the adverse events in animal trial. Because when we talk about animal trial, so there are certain condition in which some rats may goes into the fatal condition, some species of rabbits may goes into the fatal condition. So we should not stop the trial, it should be continued. So what we can do, we have to make a one catalog in which we have to make the alphabetical arrangement of the those medicine which are helpful in managing the critical or the adverse event in animal model. So this should be one of the recent advancement. Again, it is not written in any textbook of pharmacy, so that should be also included. So what we found, the arsenic album 200 with only 2 drops in a distilled water plays a major role on CNS affection, on the various adverse event in the rats. So this should be included in our catalog. So we should maintain a one catalog for animal model. That should be also important for this preclinical study. We should have a certain bunch and the what we can say the buckets of those medicine which are helpful in such conditions. Next is this is our in vitro study. We perform this study in our pharmacy college, in our PIPR, Parul Institute of Pharmacy and Research. So we develop a one culture media, homeopathic medicated culture media. So it would be a one of the recent advancement, because nowadays we have no such homeopathic medicated culture media. We have in general culture media like agar agar, blood agar, nutrient agar, growth. So these all are the culture media which are commonly used to cultivate the various aerobic and anaerobic bacilli, the gram positive and the gram negative bacilli under certain temperature in couplet method in certain culture media. So here from Parul University, it is first time we establish a homeopathic medicated culture media by using homeopathic mother tincture. So what we found that at certain concentration, suppose if we talk about Echinacea mother tincture, so this Echinacea mother tincture, it is commonly used for the uh, antifungal, antibacterial, antiseptic. But beside of this, what we found that in certain concentration and the microliter of this Echinacea mother tincture, it promotes the growth of certain bacilli and the fungus. Either they are candida or some other bacilli, aerobic, anaerobic. So this should be also important for us, because when a single that particular molecular entity of Echinacea mother tincture, when it promotes the growth of bacteria, so it also plays a major role in terms of culture media. So this is a homeopathic medicated culture media. Again, it is a very controversial part. The pharmacy people, they are not easily accepted and feasible with this type of practices, because in general they are, even though not awareing about the culture media with the modern pharmacy. So they use in general culture media. But now we introduce this Echinacea culture media with this particular variety with the oil base, and they are stimulating the growth of Candida albicans, E. coli, Staphylococcus aureus, Streptococci. So they are promoting the growth of bacteria. So this particular Echinacea culture media, it promotes all. So this is one of the novelty in our research work, because for any bacilli, different culture media has been used. Suppose if we talk about nutrient agar, so it is not feasible for all bacilli. It is not feasible for the promoting the growth of all aerobic and anaerobic bacilli. But what we found that in our particular drug entity, it is having that capacity to promote the genetic inheritance and the growth of all and all bacilli with the same concentration. So this is one of the recent advancement in our homeopathic pharmacy. We also publish it in a public domain, and this work should be this work is published in our international journal of pharmaceutical. So this is again a huge victory of our Parul University to facilitate this type of research work, even though it is not written in any textbook, in any pharmacopoeia. But again, it plays a major role in terms of commercial ground, because nowadays we have a major issue regarding with the funding. When we talk about Ayush, so H is always at the last. So the majority of fund goes to the Ayurveda, the Siddha, the Unani, naturopathy, and at the end we have homeopathy. So what we can do, we develop such type of media, we develop such type of research work, so that people can recognize the beauty of homeopathy. Afterward, we can go for the selling and the purchasing of such type of media. So it enhances the growth parameter of pharmacy, homeopathic pharmacy. So this will give you a remarkable changes in our field of homeopathy. that should be included. Now, this is our research work showcasing inhibition zone of homeopathic culture media and the fungus and the bacteria assay. And this work is completed by Dr. Sneha Agrawal, madam. She is a professor in CVM in Department of Pathology and the Microbiology. Dr. Janki Patel, she is a professor and the HOD in the Department of Pharmacology in PIPR, Parul Institute of Pharmacy and Research. And Dr. Pratibha Kani, madam, she is our professor in the Department of Homeopathic Repertory and the Case Taking in PIHR College. And she is only one who establish and prepare such type of culture media. And the Parul Patel is our trustee. So this work is already published, and you can see this research work in the public domain, and it is very fruitful for us, and it is one of the example of recent advancement. Now, the another one is the plant vaccine development. Now, we can also go for the development of certain vaccines from the plants. So, this will also give you the very conclusive and the informative results in the field of homeopathic pharmacy. So what we can do now, we prepare a culture media by the homeopathy. Now what we can do when we talk about this plant vaccine, so when we have this particular antigen candidate, so when we introduce this antigen in the bacteria or any plant material, so it gives you certain extraction value. And when we have this extraction value, and when we tested this extraction value and the edible plants or the antigen in the animal model, so this will also give you the one of the recent discovery in the sources of homeopathic pharmacy. So where is the homeopathy? The homeopathy plays major role in the selection of this particular candidate, this particular antigen, the preparation of media, the preparation of such particular bacilli and the bacteria, the culture of such particular organism. So this plays a major role. So this is one of the recent advancement in the field of homeopathic pharmacy. It is again very difficult to digest, but when we do it, so we have a certain what we can say a different orientation of researches in agro homeopathy. So we can also go for that, and this is one of the active immunity, it is not passive immunity. Because when we talk about passive immunity, so when there is a exposure of disease in any living entity, afterward there is a formation of antibodies, and after this formation of antibody, now it is said to be a passive immunity, but now it is a active immunity. Now, bioculture analysis, bioculture assay, we can also go for the animal model trial for collecting certain spleen cells and the myeloma cells. And when we fuse this different variety of cell, so they form hybridomas. And when we culture these hybridomas in a particular media, which is homeopathic medicated culture media, so this will give you the harvested monoclonal antibodies. And when we inoculate such harvest monoclonal antibodies in any living entity, so this will be also comes under the recent advancement in the bioculture assay with the homeopathy, because ultimately we prepare homeopathic culture media for hybrid, for making proliferation of such hybridomas. So this is one of the recent advancement. Now this is one of the our training session in the Parul University for SEM analysis, where different faculties interact to each other, where they share their knowledge, share their experiences, through which we have a certain modulation and faculty development program. So this is one of the training session in our Parul University for the recent advancement. Now this SEM, scanning electron microscope, this will give you the recent advancement in our homeopathic pharmacy, definitely, because now we have nanotechnology, now we have nano formulations. So when we talk about nanotechnology, nanoparticles, so definitely this SEM and TEM, which are the types of electron microscope, plays a major role. So this is again available in our Parul University, and it is easily what we can say a very feasible for all type of procedures and the solvent system for their standardization. Now recently we started with the preparation of nanoparticle by toxic human blood serum, which comes under green synthesis. So this is again one of the recent advancement through which we prepare nanoparticles by the toxic human blood serum. Now, what is the utility of this toxic human blood serum? It is used for preventing certain blood disorders. So ultimately, why we prepare nanoparticles? Because this nanoparticle having a specific action on a particular cell or the tissue. So this is one of the specificity in the nanoscience. So we enters in this particular branch. This is our nanoparticle by green synthesis, Thoja occidentalis. Now these are the images, a real image. It is not copied by any Google or any website. This is again published article from our Department of Homeopathic Pharmacy, Home Research Unit, PIHR, Parul University. So these are the nano structure and the nano formulation of green synthesis in toxic human blood serum. So this is one of the recent advancement, and that should be also included in our homeopathic pharmacy. Now this is DNA fingerprinting. Uh, when we talk about Dr. M.K. Sahni sir, so he always used to take a human biological sample like a hair from the patient and dip this biological sample in their respective medicine and give 10 downward strokes. So that energy reaches to that particular entity, living entity only. So we started this DNA fingerprinting with the hair bulb in the alcoholic base. So this is one of the recent advancement, and this research was completed with the allied sciences to complete this study and to showcase the evidence-based medicine. Thanks to Dr. M.K. Sahni sir for giving us this great opportunity and the platform to showcasing our research work in the right propagation with the hard work, with the full of dedication, etc. Now these are the changes in the carbon element in the alcohol base. After this potentization and the DNA fingerprinting, we enters into the carbon element configuration by the SEM, scanning electron microscope. So this is again one of the recent advancement. What we found, after potentization, we have two sample, one sample with the hair bulb in the alcohol, another sample without hair bulb in the alcohol. Both the sample goes under the potentization, the succussion. What we found, the hair bulb changes the configuration of carbon element. So this is one of the recent advancement. And now these are our BHMS, the MD students who started preparation of various nanoparticles in different solvent system with different technology and the method of preparation, and showcasing this research work in the international platform. So this is one of the recent research work. Now these are our first year BHMS students. Can you imagine this research work with the first year BHMS? No, it is not possible. But this research work we started with our first year students. They are not just started preparing the formulation and various external application, the nanoparticles. They also standardized it and also published this work in a international peer review index journal. These are again our second year, first year students. They analyze it, they use UV visible spectrophotometer. Now, UV visible spectrophotometer analysis is not recent advancement, but we make certain standard operative procedure and the certain methodology for determination of minute and invisible structures in terms of very negligible concentration with the different wavelength in a different solvent system. So this is again by our first year students. Now these are our PG students doing same research work with the full of honesty, dedication and the hard work. Now, she is our main pillar of microbial and the pathology regarding research work in homeopathy. Dr. Sneha Agrawal, madam. She is professor in the Department of Pathology and Microbiology, and she concludes the whole research work with the homeopathic medicated culture media. Now, Dr. Pratibha Kani, madam, she prepared homeopathic culture media with the oil base. Dr. Jayashree madam, she started showcasing of nanostructure, nanomaterial, nanoformulation, nanoparticles in polyculinum 200C. Now, this is the determination of arsenic album in the human blood serum before and after treatment by the SEM analysis. So again, it is one of the recent advancement. This is our UV machine, which is commonly used to determine the concentration. Now, this is our inauguration of OM research unit in our PIHR. So, after inauguration of this research work, we conclude many research and many articles, many patent and many formulary with the different dimension and the aspect of pharmacy. This is our faculty which involved in various research program, faculty development program to cultivate the knowledge and the practices, best practices and the communication skill in Parul University. Now this is our pedagogy session where we nourish to each other, where we facilitate to each other, where we conclude various research work with the different dimensions. Now, this is advanced teaching of MISM. Where is MISM? The Victor Ambrose and the Gary Roquin, these are the Nobel Prize winner in 2024. So they discover micro mRNA in a specific species of this worm. So this micro mRNA transmitted from one generation to another generation and carried the different memories. So this is what? This is also a MISM, the SORA, psychosis, syphilis and the tuberculosis. So that part should be also included in our research work, in our advanced teaching. Now, these are our faculties, Dr. Anand Joshi sir and Dr. Mahendra Singh Rathod, who is the main pillar of micro nano department of Parul University R&D cell, who conclude our research work, who receive our sample, who organize various workshops and the seminars for such type of research. Now this is our article publication with the different domain, with the different faculties. Thank you so much.
Okay. So the next. Thank you, Dr. Suraj Bahadia. It's really a very good session indeed. And you really showed us the beauty of pharmacy and how one need to study everything in pharmacy and what are the things that happens in pharmacy and how your college teachers as well as your friends and your students are doing research work in your college. All these things are really brilliant one. And now I am inviting those you all to participate in the discussion. And I'm inviting Dr. Ramaresh as well to say a few words on this session.
Thank you, Dr. Dhanesh ji and Dr. Manoj ji, Saila ji, and congratulations, Dr. Suraj Singh Bhadoria ji. Great presentation, research work, Parul University and college, very good research work, their drug development process, nationalization, herbal drugs, tissue, nanoparticles, potentization, very good research work. Very, very thanks, Dr. Surajji.
Now I invite Dr. Manoj to say his comments as well as the vote of thanks to the session.
Thank you, Dr. Dhanesh and Dr. Suraj. It's a commendable work, and we must appreciate and salute your studies with the university, Parul University. They are doing a great job. I think it's one of the largest campuses in India conducting the homeopathy course also. Am I right, doctor?
Yes, sir. Yes, sir.
Yes. I recently saw an advertisement this name of this university. So not only that, you have involved the junior students to this the research activities, the works. So definitely their brains will be ignited. In fact, we need the ignited minds, not the ordinary minds, because homeopathy, the study of homeopathy is not a simple thing. It's an extensive study, and also the different latest the mRNA, as you mentioned, the micro RNA particles. These are all very important. Moreover, the so-called academicians or the scholars, they are always interested in hearing such technical terms from a homeopathy physician. That will also give more mileage to homeopathy. Anyway, nice presentation. You not only participate, but also kindly, regularly attend this wonderful session for the last 1817 days continuously. Can you imagine such a work? Dr. Dhanesh and Dr. Sahan is the backbone of IFPH. So on behalf of IFPH, I'm expressing sincere thanks to all those participated, as Dr. Suraj and all the persons. Thank you.
Thank you.
Thank you. Thank you, Dr. Manoj and Dr. Suraj Singh Bhadoria. There's a big program coming on the September 28th of this month, September this year. That is just in the coming month. We have a 50th anniversary of International Forum for Promoting Homeopathy being celebrated at Taj Vivanda, Cochin. And I'm inviting all of you to register for the same, just 3,000 rupees for the same. Please kindly register it before September 1st so that you can register with 3,000 rupees, and otherwise you will have to pay 3,500 for the same. So thank you all, and tomorrow we will have another session from another professor, Dr. Dilip Bhadoria, and the subject of discussion will be knowledge of community medicine for homeopathic practice. With that, we come to the end of the day, 1817. Thank you all. Good night. So to Dr. P. Shaila to moderate the Malayalam session.
താങ്ക് യു ഡോക്ടർ ധനേഷ്. എല്ലാവർക്കും നമസ്കാരം. ഇന്ന് IFPH നടത്തിവരുന്ന മലയാളം വെബിനാറിന്റെ 1817 ദിവസം. ഇന്നത്തെ